So at this point, you should have had a knowledge of gross anatomy of the kidney, histology, and physiology. That is filtration, the mechanism of filtration, and then the regulation of filtration, and reabsorption in different parts of the tubule, as well as secretion in many parts of the tubule. We also have a knowledge of hormones and their effect on the nephron in fine-tuning the reabsorption or secretion of substances. And what we want to do here is pretty much a review of what you know, but focusing on specific ions in one key solute, urea. So let's begin the topic of how potassium ion is handled by the kidney. Let's look at the situations such as increased potassium intake, which would obviously increase the concentration of potassium ion within the blood. That triggers the release of aldosterone, which in turn would trigger the secretion of potassium ion in the late portion of the tubule, that is the collecting ducts, the cortical portion of the collecting duct. Here is additional detail that we have covered, just summarizing it here. Here is the amount of potassium ion filtered versus the amount that's reabsorbed. Please pay attention to the arrows. Anytime the arrow is out of the tubule, that is reabsorption. One in the proximal tubule, the other one is in the thick portion of the ascending limb of loop of Henle. Okay, between these two parts, you have covered about 92% or so. So now, what is happening here is the secretion of potassium, and this is, as I mentioned, in the cortical portion of the collecting duct and the very last part of the distal tubule, very end of the distal tubule. So here is where you would expect aldosterone to be able to influence the secretion of potassium ion back into the filtrate, and therefore, and that's about 4%. So we had about 92%. Now we have to subtract about 4%, so that would be 88% of potassium ion, all in all, is held in the body, and about 12% of potassium ion that was filtered is lost in the form of urea. Okay, so that's all summarized in these bulleted points. Aldosterone stimulates potassium ion secretion by increasing, so this is basically talking about the mechanism by which aldosterone brings about its effect. First of all, it adds more potassium channels. Okay, these are channels that allow potassium ion in the form of diffusion, passive movement, and also increase the activity of sodium-potassium pump, sodium-potassium exchange pump, so that there is more of potassium being moved into the cell and then sodium ion out of the cell. Now we are talking about the concentration of potassium ion within the blood versus within the cell. As you already know, most of the potassium happens to be within the cells. Intracellular concentration is the greatest for potassium ion. Extracellular concentration is only about 2% of total potassium versus 98% within the cells. So these two are simply a review. Now let's look at a specific condition such as hyperkalemia, that is greater than normal levels of potassium ion in the blood. Hyperkalemia, as you already know, it interferes with the electrical activity in the neuron, in the heart, and that may lead to cardiac arrhythmia, cardiac arrest, fibrillation, and that sort of thing. Okay. We also know that insulin triggers the uptake of potassium ion. Okay. Another important point to make a note of here is anytime there is strenuous exercise, okay, such as this physical stress, hyperkalemia, because of the release of potassium ion from skeletal muscle. Okay, so there's more potassium ion released during strenuous exercise. What that means is that leads to hyperkalemia. Increased intake, increased secretion by principal cells, and vice versa. So anytime there is increased intake of potassium ion, there is increased secretion of potassium ion by one specific type of cells, principal cells in the distal tubule in the early part of the collecting duct. Here we are looking at a couple of mechanisms that are involved. One of them is you're talking about the activity of the sodium-potassium exchange pump being enhanced. We also talk about the potassium channel. Okay. So what you're looking at is potassium ion that is brought into the cell simply diffuses out of the cell. Okay, so that is secretion. Now, ENaC, epithelial sodium channel, is what it means. All right, so you are looking at potassium ion passively leaving, sodium ion entering the cell simply because of the gradient that's set up by the sodium potassium exchange pump. So this is how you have increased reabsorption of sodium ion, increased secretion of potassium ion. All right, so that's important. Uptake of potassium ion into the principal cell by sodium potassium exchange pump, right? Diffusion of potassium into the lumen through the potassium channel is what we see. All right, so with that, we are done with potassium ion regulation. Let's look at sodium ion regulation at the kidneys. Okay, here are some factual details. Approximately 67% of reabsorption occurs at proximal tubule, and you can break it into how exactly reabsorption is done in the early proximal tubule versus late proximal tubule. All right, in the early part of the proximal tubule, there is a lot of reabsorption going on, nutrients, especially glucose and amino acids, and then phosphate and lactate. So a co-transport of these along with sodium ion. So it's making its way in the name of the transportation of other solutes, other ions. All right, counter transport via sodium hydrogen exchange. So hydrogen ion is secreted as sodium ion makes its way into the cell. This is totally, hydrogen ion is linked to the reabsorption of filtered bicarbonate, okay? A bicarbonate ion that is reabsorbed. There is a specific mechanism by which bicarbonate is handled at the proximal tubule, which we will look at in detail in the acid-base balance lecture later. But for now, just know that the hydrogen ion that is released along with bicarbonate, hydrogen ion is secreted while bicarbonate is reabsorbed. Okay, so that's where sodium hydrogen ion exchange occurs. That's what you call as countertransport. Sodium ion moving in, hydrogen ion moving out. In a late proximal tubule, note that the nutrients are already fully reabsorbed by this time. So now sodium reabsorption continues, but now the mechanism will be different. That is reabsorbed with chloride. Okay, so along with sodium ion moves into the cell, and chloride simply follows it. Okay, it's basically the opposite charges being attracted to one another. Chloride makes its way into the cell and then into the peritubular capillary eventually. All right, so that is 67% in the PCT. Now let's talk about 25% reabsorption in the thick ascending limb. All right, in the thick ascending limb, sodium ion reabsorption is again in a co-transport mechanism where you have 3 ions being reabsorbed simultaneously. I want you to see that it is 2 cations for the transportation of 2 anions. This is chloride ion, 2 chloride ions being brought into the cell along with 2 cations, that's sodium, potassium co-transporter, but some potassium ion diffuses back into lumen. Okay, so that is how it is secreted. Okay. So although it's brought into the cell, it's eventually leaving the cell. Now, but sodium ion is reabsorbed. 25% of the reabsorption occurs here in the ascending limb. Distal tubule and collecting duct together deal with another 8% of sodium reabsorption. Okay, so early DCT, early distal tubule is where it is sodium chloride co-transporter together, they move in. Late distal tubule and collecting duct is where principal cells reabsorb sodium and water. All right, so sodium ion moves in. This is all ENaC, and then water follows that. Okay, less than 1% of sodium ion is excreted. Okay, now alterations in the filtration are tubular reabsorption. Any alteration in the filtration mechanism or the tubular reabsorption would lead to increased sodium ion, increased blood pressure, increased urine output. All right, so basically any alteration can interfere with the normal reabsorption. Okay, so now let's talk about calcium ion. How is calcium ion handled by the kidneys? Well, let's talk about some details. I'm sure you already know these details, but remember, we are putting them all in a specific context, and that's where you can study each one individually. Now, proximal tubule and thick ascending limb reabsorb more than 90% of the filtered calcium ion. Okay, so that is the true detail, factual detail about calcium ion, and this is all coupled to sodium ion reabsorption. Okay, this is passive transport, co-transport, right? So now we will talk about distal tubule and collecting duct. This is where hormonal influence is needed. Reabsorb 8% by active process. This is not a passive process, it's an active process using calcium ATPase with the help of parathyroid hormone using adenylate cyclase mechanism. Okay, so this is the mechanism by which parathyroid hormone increases the rate of increases the amount of calcium being reabsorbed. Okay, so here is as another animation. If there is decreased calcium ion, there is increased parathyroid hormone. All right, which would cause many changes. Let's talk about each and every one here. Now, activation of vitamin D3 is done by parathyroid hormone. This particular detail was discussed earlier in the endocrine chapter. So you may want to visit how parathyroid hormone is involved in the activation of vitamin D3. There is an enzyme that is required in the very last step of activation of vitamin D3, and its activation is carried out in the presence of parathyroid hormone. Okay, so vitamin D3 now in its active form is able to absorb calcium ion from the small intestine. All right, increased renal calcium ion reabsorption. So that is another function of parathyroid hormone. We just discussed that here. About 8% of calcium ion is reabsorbed in the late part of the tubule. Okay, and then calcium ion release from the bones is also increased in the presence of parathyroid hormone. So all of these details are not new to you. This was discussed way back in the endocrine unit. So now let's talk about some aspects that occur without parathyroid hormone being present. So independent of parathyroid hormone, this occurs in the proximal tubule, for example, or the thick ascending limb. All right, so here you're looking at proximal tubule cells. Let's talk about it. This is the lumen, this is the interstitial fluid. This is where you would expect filtrate to be. What you're looking at is a couple of mechanisms by which calcium ion is reabsorbed. I want you to see that calcium ion may be reabsorbed by paracellular mechanism. You know why? That's because of the charges, the distribution of cation here versus cation in the interstitial fluid. It's the attraction from the more positive to more negative side, that is in the interstitial fluid. Calcium ion simply moves in the direction of interstitial fluid, and water simply follows that. All right, so here you're talking about a specific channel through which calcium ion enters. All right, so this may be a passive movement, but here is calcium ATPase. ATPase requires ATP to be able to move calcium ion from the cell into the peritubular capillary. All right, so you're looking at here sodium calcium counter exchange. For every calcium that's reabsorbed, there is three sodium ions being reabsorbed. Okay, so that is some detail without the help of parathyroid hormone. So now let's talk about calcium ion reabsorption at the loop of Henle and distal tubule, thick ascending limb of the loop of Henle, paracellular route again, and transcellular route is regulated by parathyroid hormone and vitamin D. Okay, to some extent, vitamin D also regulates the transcellular route. Okay, so parathyroid hormone promotes the reabsorption of calcium ion by activating calcium ATPase. So distal tubule is where it's dependent on the hormones, parathyroid hormone and vitamin D. Mechanism is active transport of calcium ion and counter transport that occurs between sodium ion and calcium ion. All right, so that is what we saw before, sodium ion, calcium ion. So this is the counter transport that you see in the distal tubule. All right, so with that, let's move on to talk about phosphate ion and how kidneys handle phosphate ion. Okay, 85% of filtered phosphate is reabsorbed in the proximal tubule itself. Okay, the mechanism by which it's transported is a co-transport mechanism, sodium and phosphate, both in the direction of the peritubular capillary. The mechanism that is used by phosphate, used in the reabsorption of phosphate handling is CAMP mechanism. Okay, so this is again, you're talking about parathyroid hormone mediated effect. What mechanism does parathyroid hormone use? It's the cyclic AMP mechanism. All right, so we talked about 85%. Now let's talk about 15%. The remaining 15% excreted in urine. It's not reabsorbed at all. Okay, not reabsorbed at all. It is excreted in urine. Increased phosphate, note here, increased parathyroid hormone decreases transport maximum for phosphate. Transport maximum for phosphate would be reduced so that phosphate that is in the filtrate will be eliminated, not reabsorbed. Let's talk about magnesium ion and how that is handled at the kidneys. Now, 25% of the magnesium is reabsorbed in the proximal tubule. That is regulated by parathyroid hormone. Okay, 65% at the loop of Henle. Here, please note, magnesium and calcium compete for their transportation and for their reabsorption. Okay, so if you use an example here, hypercalcemia. When there is hypercalcemia, there is increased magnesium ion excretion. Why? Because there is more calcium. Most of the time, it's calcium that is reabsorbed. So magnesium is not reabsorbed as well, and that is why it is excreted. All right, hypermagnesemia would increase calcium excretion. All right, now less than 5% of reabsorption in the later part of the tubules, such as distal collecting tubules. All right, so with this, all the ions were reviewed. All right, let's talk about urea now. Urea is a solute that is a nitrogenous waste, right? That is filtered, and 50% of the filtered urea is reabsorbed in a proximal tubule. Okay, so one interesting thing you may think about is why is urea reabsorbed? Urea is a waste. Well, what you will notice is reabsorption is one thing, and then the secretion happens elsewhere. So it's not exactly reabsorbed fully. There is some reabsorption, some secretion later on. Okay, 50% reabsorbed. So now let's talk about ADH and its influence on the handling of urea. ADH increases permeability of urea in the medullary collecting ducts. What does that mean? Well, when you have ADH, there is more water being reabsorbed. So the concentration of the filtrate would be greater than normal. Okay, at the time, what happens is urea will be released into medullary interstitial fluid just to balance the concentrations of solutes within the tubular fluid. So that's how things are affected. That is, in the presence of ADH, you would allow urea to escape from the medullary collecting duct parts. So excretion depends on urine flow rate. Remember that. Okay, if it's if there is more water being reabsorbed, there is less urine flow rate, isn't it? Less urine being released. So increased urea reabsorption. When there is less, there is more reabsorption possible. All right, and less urea excretion. They go hand in hand, correct? On the opposite side, all right, if there is less water being reabsorbed, that means no ADH, increased urine flow, all right, more fluid there is, less absorption there is, got it? So it's saturated kind of. Decreased urea reabsorption and increased urea excretion.